So yeah, as Claude said, I'm a fairly new group leader at the EMBL in Heidelberg, and I'm focused on understanding sort of the effects of temperature on zebrafish development. And we use both statistical and computational techniques alongside single-cell RNA sequencing to ask these questions. So in my lab, we're interested in this, I'll stay close, into this long-studied phenomenon wherein animals that experience an environmental stress during early development, say increased temperature, tend to generate increased phenotypic variance, so this wider red part of the plot. And the question is simple, we just want to know where this comes from. So specifically, we want to know both the molecular and cell type specific sources of this increased variability under temperature stress. And so this is a very basic science focus, but as we've seen, especially from the talks this morning, this is becoming more and more practically relevant as rising water temperature solidifies into reality here. So what do zebrafish look like when they're raised at high temperature? So these are a couple normal fish, 2-day-old zebrafish embryos, raised at their preferred temperature. And if you were to increase the temperature just a few degrees, you would see something like this. So about, let's say, 15–20% of the embryos will have this bent tail phenotype. And others have noted several other consistent developmental defects at high temperature. And in addition to this sort of variable phenotype, we know that temperature increases developmental rate. This is probably intuitive to most everyone here. So these fish will meet their developmental milestones more quickly than the ones raised at 28 degrees. So we've got these two features of environmental stress, this variable phenotype, but also this increased developmental rate. And we want to know the cell type specific sources of these effects, so we're going to use single-cell genomics, but we're going to need to adapt it to account for these things. So first of all, single-cell genomics, which we've heard about a couple times here, but I just give a really brief introduction. So if you want to know what's happening in different cell types of the animal, this is sort of the premier approach to do this. And the technique is very simple, and I just want to have you understand what the data look like. So you dissociate an embryo, and inside these cells you pull out RNAs, and because there are so many activated genes in each cell, you get this really rich high-dimensional data. So in the case of zebrafish, about 30,000 rows in this matrix of genes. And using a kind of machine learning approach, you can view the relationships between all of these cells, the columns in the matrix, in 2 dimensions. So this is the most common view. We've already seen a couple of these. So in these maps, each point is a cell, right? And they're similar if they have similar, they're close if they have similar transcriptomes. So this work here actually represents a kind of state of the art. This is from Alex Shear's lab, and was the first instance of this technique being used in fish development. And it's amazing, but the one thing that it doesn't have, despite all of this high cellular resolution, is the ability to resolve individual embryos. So I told you we had these variable phenotypes across individuals, and so we need our single-cell technique to account for that. So almost all the experiments like this would pool many, many embryos together and profile them at once. So a lot of cells, but only one sample. Now, so we had to adapt this to make it work for individual fish. And so the exact technical details aren't really important for this talk, but I'm just going to show you the trick that makes it all work. So we take fish embryos in wells of a 96-well plate, and we dissociate them as normal. And when we lyse the cells and get the nuclei out, we add this little, there we go, this DNA oligo, this hash as we call it, and it has a barcode that's specific to the well, and it also has this poly-A sequence. So in the end, whenever, whatever single-cell platform you use, you'll recover these barcodes just like you will all the transcripts from these cells. And if there's time in the end, maybe we can talk about it, but the approach we use to generate single-cell transcriptomes is not based on droplet encapsulation like in 10x, but it's this single-cell combinatorial indexing approach. And I'm going to skip all of the details other than to say what happens in the end is we barcode all of these transcripts with kind of a multi-layered barcoding scheme that tells us what cell did it come from. And furthermore, for every cell, we can say what embryo did it come from. That's what's new. Okay. So for the first time, you can kind of see the reproducibility of development. We can get replicates. So we use this approach to make an atlas of temperature stress during the first couple days of zebrafish development, looking at a few hundred embryos at these 3 different temperatures. And one important thing to note about these temperatures is that None of them actually crosses the threshold into a full-on heat shock for zebrafish. This is warm water, increasingly warm water. So as before, these points represent cells, and I've labeled some of the broad tissue groups. So together, this data set has about 500,000 cells. And with this, we can now start to unpack, how does each cell type respond to temperature, and whether some of them are more sensitive than others. Now, to make sense of the effects on temperature, we also need a control comparison at normal temperature, so we also built a comprehensive atlas of zebrafish development. So this is actually perfect. Exactly where Sanjeev's video dropped off is the first time point in our data set here. So there are about 18 time points sampled together, comprising about 1,000 individual embryos and over 1 million cells. So we think we have very high cellular coverage of zebrafish through organogenesis. And yeah, this just gives you a visual of the total range of the time frame. Okay, and the last thing I wanted to highlight is, for some of these cell types, you can see the quintessential developmental trajectory. So these cells are colored by time point, so they lay themselves out in accordance of their developmental stage from early to late. So this is a nice feature of the data that we can use. So this data was very painstakingly annotated, and I'm showing all of these annotations here. And this together is about 85 broad cell types and 150 or so cell subtypes. So I'm going to pause for 1 second just to highlight a quick important computational technique for analyzing the effects of temperature. So you may have noticed that our temperature map looked a lot like the reference map, and that's not an accident. So we use a computational approach that's increasingly popular in the single-cell field, which is we can go from this reference data and we can extract from it the underlying models to build this representation, which in our case is a linear dimensionality reduction, about 100 principal components, and additionally the nonlinear visualization with UMAP. So you can pull out these models from the reference, and when you get new data, you can simply transform it and project it into the same space. And yes, so you end up getting a map that looks very similar, but importantly, you can transfer all of the labels, say your painstakingly annotated cell annotations, to your new data set without doing all of that again. And if any of you have ever done that, you know that's a huge benefit. So, coming back to the data, the last thing I want to highlight that's unique, in case you missed it, is that underneath this big map of 1 million cells, there are little maps for every individual embryo in the data set. So that's what's new about this data. Now, the result of doing that is a completely new data structure that contains both the standard single-cell RNA-seq representation, genes by cells, but now since we know the embryo of origin for every cell, we can group them and make this very useful abstraction. So a new matrix of cell types by embryos. And so the values in here now represent something like anatomy or cell type composition. So just going to give an example now of why that's actually useful to have this data. So here it is, same data as before. It's our cell type by embryo matrix, and we can more or less treat it exactly like the single cell data, process it with the same tools, and we can make the same types of maps. So now, the big difference is each point in this map is not an individual cell, but rather an individual embryo. And embryos will group together on this map if they have a similar cell composition or anatomy. And what hopefully you can see is when you color all of these embryos by the time point in which they are sampled, that more or less defines exactly this trajectory from early to late. Now this is useful because the algorithm here doesn't know anything about the different ages of the fish. This just gives us a way to kind of measure it from the data itself. And that's important because it gives us an opportunity to precisely account for one of the effects of temperature on development, which is to increase the developmental rate. So let me show you what I mean. So if we take now the same map and we just pull out one time point, 24 hours, post-fertilization, and then we highlight the temperature that each embryo was raised, you can see that the high temperature embryos are always creeping a little bit ahead on this trajectory. And that's true for all of the time points we test. So that's not surprising in itself, but what's important about this is for the first time we can give a quantity to the developmental rate acceleration. So not only can we account for the effects of developmental stage when we build models to understand the effects of temperature, we can also capture variability just in wild-type developmental stage. So, when you fertilize fish, they're not fertilized in the exact same moment. There's subtle variations in embryogenesis, and you actually end up with a series of embryos that are at slightly different developmental stages, and for the first time, we can account for that. So when we do statistics, we can include it as a covariate in the model. And this concept is identical to the concept of pseudotime, which maybe some people have seen in single-cell data, but more or less position along this trajectory is what we're using as a latent variable for developmental stage. Okay. So we have this new way to capture embryo stage straight from the data, but what about something new? So we wanted to know if some of this variability we see in phenotype comes from non-uniform acceleration across different cell types. So how would you measure something like this? We go back to our trajectories now of cells. So we can take, for example, a single tissue like fast muscle, and since we know that sort of the early myoblasts are over here and the differentiated muscle fibers over here, we can draw one of these trajectories. And just as in the whole embryo, fast muscle cells from high temperature fish will be a little bit accelerated on their path in this trajectory. So because we can compute a kind of, we can use this concept to compute a kind of transcriptional age of every cell type. And that again uses a similar concept to our label transfer procedure before. We just go to every cell, say in our high temperature experiment, ask what is the time point of all the neighbors in the reference data, and you can just take the average of those time points. So if temperature is scooting them ahead, you'll detect that. So this is what it looks like when you compute these cell type specific ages for all the cell types in the embryo. So this is just one time point now and all the different cell types here on the x-axis. And so at high temperature, we see something interesting, which is that, of course, cell types accelerate. Now they're ordered here by their relative acceleration. So both at 32 and 34, you see these cell types that are really, really speeding ahead compared to the rest. And beyond that, you even see a few that don't do much of anything at all in response to this temperature treatment. So what that tells us is that, yes, whole animal stage is increased, but this developmental acceleration is non-uniform across cell types. So potentially a source of variability. Another question you can ask, same concept, but you can go one layer deeper, is you can ask about synchrony or coordination between the cell types. So, does temperature cause all of the cell types in the fish to accelerate in an equal coordinated fashion? So because we have, for every single embryo, the age of every cell type, we can start to do things like this. So for example, we can look at, here's our muscle cells again, so y-axis is muscle cell age. Now we can compare it to another cell type, say the notochord. And if we do this across lots of many individual embryos, we can see that, yes, as the muscle age increases, so does the notochord. And so if this correlation is positive, then these cell types are coordinated. And yeah, you would expect most cell types are coordinated. And thankfully, that's what you see. So this is the normal temperature. And this is a heat map showing the kind of pairwise correlation between every cell type in the fish with every other cell type. And it's very red, which means things are very coordinated. However, at high temperature, you see a lot of this correlation structure is lost. So these are ordered in the same way. And that's even more obvious if you just look at kind of a distribution of these correlation coefficients. So using this technique, we found, which I think is a kind of hard to measure phenotype, this coordination, and we've showed that temperature stress actually reduces coordination or synchrony among cell types. Okay, so now that's the part on developmental rate. So now I want to say a little bit about, that was a very abstract phenotype, I would say. And so what about the more straightforward phenotypes of just the cell type compositions of these animals? So once again, like getting a matrix like this is fairly hard with any other method that I'm aware of. So getting counts for each embryo is useful to see, are there some cell types that are lower or higher in number in these high temperature embryos? And of course, because we don't just have an N of 1, we can do real statistics. And I told you that we can account for developmental stage in our models. And what this lets us do is look at for each cell type. So again, we can account for embryo stage, and we can test for effects on the mean cell abundance, but also the variability to see if we ever see any effects, kind of like the thing I showed you at the beginning of the talk, this increased variance. And this whole analysis kind of comes down to addressing a common assumption in zebrafish temperature treatments, which is that high temperature embryos are really just like wild type, but ahead in developmental age. And I would argue just the presence of some of these anomalous phenotypes suggests that's not the case, but we can test it now. So this is the result. So this map, maybe I can use this screen too. This heat map, every row is a cell type, and the one on the left shows effects on the mean. So this means red means that cell type went up, blue means the cell type went down in abundance, and on the right we see the effects on the variance. So shown here is a coefficient of variation. And what I hope you can see is that after we account for developmental stage and isolate the effects of temperature on the animal, we can see that the composition truly does change. So some cell types are reduced in number in these fish. They're not the same. And to move on, I'm just going to give an example of how these new phenotypes have led us to understand the kind of molecular source of temperature sensitivity in the notochord, which is a cell type where we saw both a decreased mean and an increase in variance. So again, mirroring that effect from the beginning of the talk. All right, so the notochord. Probably doesn't need introduction in a room full of chordates, but this is very important structure, structurally and for signaling during early development in vertebrates, and it has two main cell types that you should know about. There are these so-called vacuolated cells. They puff up like a balloon in the middle of this structure, and there's this epithelial sheath, there's the epithelial sheath cells, which themselves secrete the epithelial sheath here, that you can see in this EM image. So if we isolate just now the notochord cells from the data, we can start to see, what is the source of our temperature sensitivity? Is it these vacuolated cells or is it these sheath cells? So if we isolate now just the notochord cells, they're here colored by time point, early to late. I hope you can see that the kind of trajectories to each cell type are very, very clear. And you can see that, of course, there are many more sheath cells than there are vacuolated cells. And I don't have time to explain fully this analysis, but we did some spatial statistics that would be more common in demography or political analysis to find areas of this map where these high temperature notochord cells were kind of biased in their transcriptional state. And what we found was the biased area of the map was entirely among the sheath cells and not the vacuolated cells. So this tells us that this temperature sensitivity is probably coming from this cell type. Secondly, and it's nice that these data also have transcriptomes underneath. We then can go in and ask what's actually happening in these cells compared to the rest of the sheath cells. And what they're doing is upregulating translation. So it's a lot of protein synthesis, which makes sense. This is a cell type that secretes collagen to make this sheath. And the other thing it's upregulating is the unfolded protein response. And that also, in a way, makes sense. But it's a little surprising because it's a little paradoxical because UPR is usually activated to turn down translation. If you have an unfolded protein problem, you want to stop making protein, get it figured out, and then go on ahead. These cells are doing both. So probably not a good idea. And so I just wanted to know what happens to these cells if you take away their ability to activate this unfolded protein response. Possibly, yeah. So more or less asking, how would these cells respond without the ER chaperones? Oh, I didn't mention this, I'm so sorry. This is a process that's fully entirely in the endoplasmic reticulum. Again, we're talking about protein secretion here, sorry about that. So the question is, for the ER chaperones that are upregulated by the unfolded protein response, how do these cells respond without? And it was actually really awesome. I saw that someone had made a similar mutant on a poster today. So ATF6 is a transcription factor that both senses and then activates this unfolded protein response. And yes, much like I saw on that poster, the ATF6 mutants don't have much of a phenotype at the normal temperature, but when you raise them at high temperature, so these are stained notochords of tails of zebrafish, at high temperature, the bend in their tail is even more severe. So the notochord seems to have some structural issues. And I actually went one layer further to look inside these sheath cells with EM, and I'm going to try to show you what I saw. This is now just in the high temperature ATF6 mutants. So this is the sheath I told you about that they secrete. This is one sheath cell. Here's one vacuolated cell underneath. And what was interesting here is we saw that these cells apparently are upregulating a lot of translation, having problems getting their protein folded. They're actually accumulating collagen fibers inside the cells, which hopefully you can see. kind of mark them with these asterisks. So there seems to be some sort of defect in secreting these outside the cell, and we think this leads to the kind of issues of this cell type in performing its function at high temperature. Oops. Okay. So now I'm going to summarize and then tell you a little bit about what my lab is kind of headed toward in the future. So I've showed you that we have this new method for systematic single-cell profiling of individual animals and how that lets you, do statistics with single-cell genomics, which is nice. I showed you that temperature stress increases asynchrony in development in addition to its more well-known effect of increasing developmental rate. And I told you that the stress sensitivity of the notochord seems to be associated with a really cell type specific demand on proteostasis. So the proper folding and synthesis of proteins. And in this case, it's because the cell type seems to have a strong demand for secretion. Okay. So this question is really, really interesting to me, and we just saw this in one model system, and we are going to follow up and figure out why it is some cell types are sensitized to temperature while others are less so. But the other approach for doing this is just to go out there, find animals that have different kind of thermal sensitivities, and then just use these same methods to address if there are any differences. So, for example, there are just a few fish here that I'm showing, and they're associated embryonic thermal limits. So zebrafish is a tropical freshwater fish. It can handle, probably on the low end, I wouldn't go lower than 20. High end, I wouldn't go above 34. There are other species, like sturgeon, that have pretty much the same range, but the whole thing is shifted down, where now zebrafish's low end is kind of like this species' high end. And there are also species like Medaka that seem to really win in that they've increased the range, so having both a higher upper thermal limit on embryonic development and lower limit than zebrafish. So we're interested in these sources of these different phenotypes, and we're planning now to build atlases, much like the one I showed you, for temperature and the normal, for both normal and elevated temperature, to figure out which cell types might be responsible for these differences. And the advantage of doing a kind of cross-species data integration is that we can associate not only, transcription levels, but also developmental rate, as I pointed out, which differs at high temperature, and also the cell composition of these animals, if that has anything to do with their resilience to high temperature. And to that end, I'll just show one tiny bit of data. So Sophie in my group has been raising these Medaka fish at high temperature, the same high temperature we raise zebrafish at. And in zebrafish, we would usually see, quarter to half of the fish would be bent at this temperature. In Medaka, it really is true that this was like 1 in 50. It's not a very common phenotype. These guys do seem to handle high temperature a little bit better, at least in the expression of this phenotype of this axial defect I pointed out. So we've now been building these individually resolved, single-cell atlases for Medaka, and it's an effective technique because now we can quickly count all the cell types, and we do think we can use some of the dataset integration techniques we used for our temperature to zebrafish reference, also across species to transfer time labels and things like that, which is of course important because a lot of species develop at different rates. So notably Medaka are at least twice as slow as zebrafish. And with that, we didn't even make it to peace. Perfect. With that, I would thank my group at the EMBL Heidelberg, and we're definitely looking for a postdoc, so please reach out if you're interested in this kind of work, and also collaborators in Seattle, as well as other core facilities at EMBL that made this whole thing easier. So, thanks.